Okay, so you may have seen, so Röntgen truly discovered X-rays. That was just sort of around the world, people were trying to understand what is an electron. This is the late 1800s. They didn't understand the electron at that point. J.J. Thomson got the Nobel Prize. I think he did his work in 1899, as I remember, and he got the Nobel Prize in something like '06 or something. But at any rate, they did not understand what an electron was. They knew what electricity was, but they didn't know what electrons were. I'm sorry, yeah, they didn't know what electrons were. So they all had these discharge tubes where they would have a cathode and maybe an extraction grid and a voltage on an anode far away, 30 centimeters away. And they would run electrons into different anodes, and but they were mostly interested in the gas discharge in the middle, and hoping that that was going to help them figure out what the electrons were. And Röntgen happened to see some fluorescence nearby, near the table where he had the discharge tube. I don't remember what it was that was fluorescing, but some material that we would commonly understand now to be a fluorescer. And he didn't understand how that was happening. How was something getting from his tube to there? And he started blocking it with different things. And what we understand now of the high Z materials blocked it well and low Z materials didn't. Reflection didn't seem to work. He shut himself in his lab for something like more than a week, as I recall. The only one who was allowed in was his wife, not even the technician. And he lived in that room for at least a week. And he wrote these really great papers by the classical physicist about what did he observe. So it was really great. And there are first images of a hand, which are not as good as this. The first images was his wife, Frau Röntgen. And they're kind of blurry, but it's like the first experiment you ever do, it's never quite perfect, right? And when he did that, I mean, it's just a shadowgram. He realized what was going on there, and he was recording it on film because actually, for sufficient energy, film works well for X-rays just as well as it does for light. And so he did that, and he formed the first images. And when he made his first public address, which is this date in January, the host of the sort of colloquium he gave in the physics department, he took that, he made an X-ray image of that man's hand. And by this time, he knows how to do it well, no blurring, no motion. And he gets his really fine images. And that's why I've chosen that one. Okay. And so that was in January. The word was not only got out, was sent out to friends by not only him, but by others, not only by Röntgen, but by others who were in the room about this amazing discovery. And they went to Europe and the United States, and people all around the world had these discharge tubes. Physicists around the world were trying to understand what the electron was. And so within weeks, they were making images like this. And in no time, well, it says medical applications in February. So people were already realizing that they could see broken bones and stuff like that. So there was an extremely rapid spread of information around the world. Okay. So that's just what we call shadow casting. Okay. And we still do shadow casting. I'll show you some examples later of micron scale shadow casting, micron resolution. To get a little enhancement of edges in X-ray imaging, a couple of techniques are used. One is called propagation-based imaging. That's what's shown here. And that is that if you make a shadowgram of an object, but you get the geometry right, the distances right, diffraction from the edges can produce little diffraction patterns. And you say, well, diffraction requires interference. They were using X-ray sources. They're not coherent. They're not coherent, but they were, if you're looking at the edges, that's only a small angle. So the large size and the finite wavelength, the d dot theta like lambda works over a very small angle, and can produce these interference rings at the edge, if the edge is sharp enough. So that's called propagation-based imaging, and people do it. And here's a diffraction-enhanced imaging, and these are images. This is a pig's eye, okay, and you can see all kinds of details. And this is just there's no optics involved here, okay. So, and they only need spatial coherence around the edges. They use a crystal in this work, crystal analyzer, okay? And here's someone's thumb, human thumb, with tungsten K alpha. So this is just an electron impact tube, okay? Just like they were using, but without the gas, okay? As Roentgen used, but without the gas. And you can see, here's the bone, here's the flesh. You can see the nail and the thumb. So these are techniques that people use without any optics, and they produce fine images that get published a lot in medical journals, okay? There's also what's called grading-based imaging, and this is also using clever degrees of coherence is introduced. So here's a double grading case, X-ray source, finite size X-ray source, electron impact tube, some object that's going to rotate because they're going to do tomography, with a grading next to it with a period D, okay? And then another grading, an absorption. So this is a phase grading. So it produces nice orders, plus and minus, no straight through, because that only comes when you have absorption. But with a good phase grading, meaning the thickness is just right for that wavelength, then you can produce two orders, actually other orders, but you'll certainly produce two strong orders, okay? And they interfere. And because they're each going off at a different angle, when they interfere, they don't interfere at D, but they interfere with the periodicity D/2. So you just have to make a diagram to convince yourself that that's true. But here they make it in an absorption grading. So the absorption grading period just matches the interference from these, from the +1 and -1 order from this structure, whatever it is. And then they shift the grading, the absorption grading, through that pattern, and they record all of that. And as a result, they get these different images. And it's minimal coherence. It turns out the spatial coherence in this case is set by the, not the size of the source, but the size of the opening in the grading. And again, small angle, small wavelength, etc. Okay, so you get this interference pattern, spatial coherence, but it's just locally, it's only coherent locally, but that's all you need to match up with one of the bars of the absorption grading. Okay, and so these are the kind of patterns that people get, and that's called grading-based imaging. And people are trying to make clinical tools with this to go into hospitals or veterinarians, but hospitals, in fact, I shouldn't say it because we're recording all of this to YouTube, but I have recently seen some really nice proposals for using this method in clinical applications in a hospital. Okay, so those are some of the techniques that are used. This is an example. It's called 3 grading thing where they produce these interference patterns, and this is by Franz Pfeiffer, now at the Technical University of Munich, but I think this was done when he was at the Swiss Light Source. So now he's at the Technical University of Munich, and this is a small fish. It's not very big. I forget how big it is. Maybe there's a scale here. I don't see it. But as I recall, oh, here's the scale, 2 millimeters. So maybe a few centimeters, 3 centimeters. The exposure time is kind of long, but they make it work. And you can see a lot of fine detail.
Professor?
Yeah.
I just want to know, can you go back to the slide where you had the crystal for the angular analyzer? And I just had the question in terms of the thickness. How is that selected
Oh,
and
okay.
and
Yeah,
then
so this is a
material?
pig's eye. It's not much different in size than a human eye. Okay, so it's what they've got is hard X-rays. In fact, here it says 40 kilovolt bending magnet radiation at Brookhaven, NSLS. Okay, and that team is now in Canada at Saskatchewan, where the Canadian light source is. But at any rate, so this is maybe 2 centimeters across, but they're using 40 kilovolt X-rays to get through there. So that's a good match for the absorption. Not a lot of absorption at 40 kilovolts. It's all phase effects at that point. Um, and we would say, well, this is a really short wavelength. So to get this interference effect at that very short wavelength, well, um, they use a crystal analyzer, like a crystal monochromator, probably a
Okay.
double crystal monochromator. And this is the angle, acceptance angle of the crystal. Okay.
Okay, got it.
Okay. So if the angle was bigger, you wouldn't see the interference effects. Okay, good. Thanks for the question. Okay, so now we're going to switch to zone plates. Zone plates for nanoscale imaging. So they've had a good life, several decades, and they're still going strong. And the resolution that labs around the world get now, the best labs, get spatial resolutions just below 10 nanometers, around 10 nanometers, a little bit better for two-dimensional imaging. Okay, three-dimensional imaging, tomography, they're limited not by the zone plate, but in fact by the depth of focus. That the depth of focus at these resolutions and wavelengths is only probably half the thickness of a cell of some kind. Okay, so these zone plates are used. And just to remind you, we had said last time that the resolution with one of these microscopes. So this is like your high school biology microscope, right? This is backlit by a white light is your sample. This would be vertical. There'd be a high numerical aperture lens microscope objective that would be kind of close, and then there'd be a very high there'd be a magnification, and maybe this distance in your high school microscope would be like 6 inches or something like that. So this is a little different. These typically run at high magnification, 2000. So this X-ray CCD would be pulled way, way, way back. And the reason for that is that the resolution of the pixel size in the CCDs are kind of big. And so you need the magnification. The resolution, if you do it right, is set by the outer zone width, which would be something like a little bit more than around 10 nanometers, let's say. Okay. And these things have the pixel sizes are like 10 microns. So 10 nanometers, 10 microns, you need a magnification of 1,000, maybe 2,000 to get a little advantage on that. This one is called a full field microscope. It images all at once this entire sample that's been backlit. It images the whole thing all at once onto a CCD, and your exposure times are seconds. Okay. The second kind of microscope is a so-called scanning microscope. Sometimes they call them STIXM. What is that? X-ray transmission microscope. So I guess it's soft X-ray transmission microscope, because both of these work best with soft X-rays. And the reason for that is how thick the zones have to be. When you try to go to hard X-rays, which I'll mention in a moment, the thickness of the zones has to be quite a bit deeper in order to either provide absorption or phase, whichever it is. The STIXM requires coherent illumination. It focuses light down to a small spot, and then you raster scan the sample in front of it and record just with a one-dimensional detector, not a CCD. So you just have X-rays coming through. This is just to block the other orders. You only want one order to come through. So it's a big hole. Allows the focused radiation to come through, and you scan. And you, as you scan in both directions, you just record that on a detector. So because this requires coherent illumination, the exposure times are much greater. I'll give you an example later. The big advantage is there's a reduced radiation dose in this microscope compared to this one. In this one, remember the zone plates had an efficiency of something like 10%, maybe even 5% when all is said and done. So let's say 10%, that means the radiation damage to this sample is going to be 10 times higher just because you have the inefficiency of this lens. So as a result, you need to do cryofixation, okay? For biological or any soft samples, you need to do a cryofixation to protect it from the radiation damage, at least at the spatial resolution that you can see. If you could see down to the molecular level, the radiation damage would still be there, waiting for you. But at 10 nanometers, we don't see that. So this one has the efficiency of the lens between the sample and the detector. This one does not. This one, the inefficiency of the lens is upstream, so that all the photons that get through to your sample are going to be recorded or absorbed, okay? So those are the two microscopes, and we'll give examples of both. And this one is, today, they are typically used for resolutions in the same ballpark, sort of 10 to 20 nanometers, but with high spectral resolution. So they'll use it, for instance, in environmental work, where they'll do an image, but then they'll scan to another wavelength, very close wavelength, and they'll get spectral scans, high resolution spectral scans, and they'll see within this environmental sample, excuse me, what's the, what is the phrase? What is the, for all of the atoms in the molecules, what state are they in, okay? So we'll see that in just a moment. So here's the full field microscope. This is the one that's at the ALS. There's ALS bending magnet. There's a plane mirror just to block high energy photons, because you're going to image here in the soft X-ray region. So you don't want any hard X-ray radiation to come through and hit your sample and produce even worse damage. So you block radiation higher than the photon energies you want. So, and normally with these microscopes, they operate from, let's say, the carbon edge up to, let's say, iron or cobalt or something like that. So under a kilovolt, okay? Here's the microscope here with the high magnification. So as a high resolution zone plate with very small outer zones, again, of order 10 or so nanometers, imaging this object onto this CCD. This is just the back illumination. So this was the high school microscope. This is the white light bulb. Here, it's not a white light bulb. We actually use a condenser zone plate, which does not have such fine zones, okay, to back illuminate the entire object. And then this thing, this zone plate does a point-by-point image by interference, okay? But every different point is incoherent compared to points nearby, but for the image produced by this microscope, over a field of about equal to the resolution, you get coherent interference. And so it works on interference, okay? The sample holder is indexed, so you can look at it in a microscope. So for instance, the biologist could go in and have a sample with thousands of cells and pick out, it could have many thousands, but they could pick out 1,000 cells, some that look just like what they think it should look like, and some that look like they're damaged or something odd about them. And they'll pick the coordinates of that because they have this indexing. They pick the coordinates, look at it with the visible light. Every time they see something they like, click on it. The coordinates are recorded, and then the X-rays are turned on, and they step through all those spots, producing image after image. So a graduate student a long time ago brought the world from taking not so many images a day to doing something like 1,000 or 2,000 images a day. Okay. the biological images and others that have to be wet are typically imaged in the so-called water window, where absorption is low, absorption is low due to the water, so it has a high transmission. But for proteins that contain carbon, nitrogen, etc., those elements do the absorption. So in this window, there's not much absorption by the water, but there is a lot by the carbonaceous material. And so there's a good contrast here. And that's what's used for natural contrast. So this would be as compared to electron imaging. The electron imaging would have far higher resolution, but it wouldn't have this elemental capability of looking, for instance, in the water window without seeing the water. What people learned, many of us learned around the same time, was that the radiation damage would prevent you from doing biological imaging in this window at this resolution. Radiation damage would prevent it. So we used to, for instance, we were looking, some group, what is her name? McGowan. Kathy McGowan was a biologist interested in malaria, malarial infected red blood cells. And we imaged a lot of red blood cells, but what we found was that at high resolution, they'd burst. We'd only get to take one image. And so the radiation damage was killing us. And friends in Germany and elsewhere were learning the same thing. And they developed this quick freeze, which the biologists were already using for electron microscopy, a quick freeze. So that was used also. And the quick freeze, at least it prevented you from seeing damage at the few nanometer level. Certainly the damage was there at the molecular level, but with our resolution, we couldn't see it. Everything was locked into place by this quick freeze. So for example, here's an image of, this is a single cell, okay? This is the nucleus, sorry. This is the single cell, and this is connections to nearby cells, okay? And these are little, so this is the nucleus and some of the nuclear material. And here, out here, there are these vesicles that carry proteins or enzymes around the cell. And so this was one of the high quality images that was seen some years ago, early days of the ALS, and using cryomicroscopy. And Carolyn Larabelle, who's both at UC San Francisco and LBL, she has a microscope. She was using the so-called XM1 microscope. That's where this was at the time, which was developed by Verna Maya-Ilse. But you'll see other stuff. So she and her husband started their own microscope program at the ALS. And this is also from Carolyn and Verna. Okay, where in this case, they labeled specific proteins. So these are structural proteins. I forget, it says it's an epithelial, hydrated mouse epithelial cell, just inside the water window at 520 EV. Okay, and there's silver enhanced gold labeling on this microtubule network. So these are structural proteins, and that's what they want to see. So the biologists would say, oh, isn't that sweet? We've seen it for decades with electron microscopy, ho-hum. But looking at specific structural proteins by labeling, that would be much more interesting. Okay. And this is work from Desi, Berlin. So they are the leaders now in soft X-ray microscopy. And they have been from the beginning, but they continue now. The biological images, I'm leaving to Hannah, so she'll present some of those images with X-ray tomography. But that same group is also looking here, because this is material science. These are looking at titanium containing nanoribbons, and they're looking at as a function of photon energy, as they tune that, what do the images look like and how do these structures change? And from that, they were able to, not the X-ray microscopy people, but these people, Carolyn, I think, Bittencourt, looking at the the spectral signatures of the different things. And they also saw certain polarization effects and everything. So I put that in. There's a reference down here, just so people in MSE would see some material science application of the X-ray microscopy. Also for civil engineering, there was a lot of work done early on about cement and concrete formation. And the study was, how do dams crack? So giant dams, hundreds of feet tall, and they were taking and actually forming, getting the cement and wetting it, the powder, and watching the formation of concrete on a really small scale, appropriate for the zone plate microscope, and seeing how the material developed and how cracks appeared on the microscopic scale. So that was also for civil engineering. Oh, here it is. I forgot I had it in. So this is the work for Professor Monteiro in civil and environmental engineering, Paulo Monteiro. And in this case, he's pointing out that the formation of cement is one of the biggest pollution problems we have in the world, producing CO2. And one of his goals in life is to find new processes, new cements that don't produce as much pollution. Oh, this is some of his examples. And yeah, of different things. So, okay, the scanning transmission, scanning, not soft egg, scanning transmission X-ray microscope, STIXM. This is the one I showed you before. It's just a sketch of it. So there's an undulator, a grating to choose the wavelength, an exit slit for this to make it a monochromator, a pinhole to set the spatial coherence, launch a spherical wave, the zone plate to focus, the order sorting aperture isn't shown, and then the detector, that the sample gets scanned in a 2D stage, and the detector records the signal. And requires spatial coherence. So the exposure times are quite long, many minutes, many minutes, but with this new diffraction limited, so-called diffraction limited storage rings, where they're going to collapse new electron optics is going to collapse this elliptical illumination down to almost a circle. At the ALS, they hope it will be a circle. Harder X-ray machines, maybe a 3-to-1 ellipse. But anyway, this is a very common and important tool to environmental scientists, among others. And the exposure times, which now are a little bit long, are going to get reduced. For instance, the one with a particular undulator at the ALS and the ellipse, they're going to reduce the data taking time by a factor of 100. So it's going to be a gigantic improvement. There's a new imaging technique called tachography, which I'm going to mention in a moment, where rather than focus down to the smallest spot and then raster scan the sample in front of that spot, they use a slightly defocused beam. Okay, and they call that tachography, and I think we'll have time to tell you what it is. This is an environmental, part of an environmental study by Adam Hitchcock and one of his colleagues. And here at the ALS, this man, Tolek. So what Tolek Tolusichek, what he's famous for is he made the scanning microscope a precision tool. Up to that time, the scanning wasn't good enough for the resolution they could get optically. It was just too noisy and bumpy and everything else. And so along came Tolek at the ALS, and now everybody uses it around the world. And he showed how you really cut down the vibrations. And he did everything. Even the fans in the roof at the ALS had to be turned off when he was trying to get the highest resolution images. So spectroscopically, they'd look at various elements in their sample. So this is a wet sample from a river, does it say? Saskatoon River in Canada. And they're looking at, in this case, iron to get the valence states of the iron. And apparently in one state, it's environmentally not good, and in the other case, it is. So they're looking at iron 2p energies around 700 eV. That's where the absorption edge is. And so this is work that is really done big time in many synchrotrons. And this is just more of the same team, Adam Hitchcock and other colleagues. Okay. So then X-ray tomography. So we're going to have some presentations on X-ray tomography. So I'm not going to speak about it here, well, except to show just some examples of it, but you're going to see more next week. But what you do is you rotate your sample and you look at different angles, and by cleverly combining what you've seen at all different angles. I mean, you can imagine just looking at 90 degrees, if you just had two views, you'd be able to say something about the location of, let's say, X-ray absorbing features in your object. But the best thing is to view this like every degree or every 2 degrees or something like that, and numerically reconstruct what your object looks like in here. Okay. And so what we used to call CAT scans, okay, CT scans now. Here's an example of a 3-dimensional image of what's called a via. So a computer chip has many layers, maybe 20 different layers. And between layers, they have to have current going from one layer up to the next. And they run the current there through little copper or tungsten, in this case, tungsten plugs, they call them, which just electrically connect one layer with the next. But as the computer chips were getting smaller and smaller and the currents were higher and higher, they found that the electrons would actually knock some of the atoms out of the way and produce a hole, a physical hole. The copper plug would have a big hole where there was no copper atoms. They had all been actually pushed out of the way by the electrons coming through in the electron current, and they would break down. The chips would break down. So these people, the graduate student, G.C. Tin, and his faculty advisor, Mao Zedong, at the Taiwan Light Source, did this images, 3-dimensional images, which was important because people were trying to understand and didn't have a 3-dimensional picture of how this happened and how it progressed in time. They could do this as a function of time. So that was that was done through tomography. Okay, and here are some other cases. This is at the ALS. So here's that same microscope we were looking at before. Does it say it's called XM1? No, it doesn't. So I told you that the German group was the leader at the time. Gunther Schmoll was the early leader, and Gerd Schneider is now at Berkeley in Berlin. And Gerd, after doing his habilitation in Germany, came to work at the ALS for like 2 years or something like that. And now he's in charge of the program back in Berlin. And what he brought us was an ability to put biological cells in a capillary, a transmissive capillary, put it into the microscope where the sample should go, and then rotate the capillary and take images. So to produce nanotomography, okay? And so in principle, the resolution would be set by the outer zone width of this lens. But in fact, it turns out that the thickness of these objects is bigger than the depth of field. So you wind up compromising a bit. And let me just see, does this, sorry. Well, I guess I didn't bring I didn't bring the movie. I didn't load the movie, but this thing rotates then, and you can see some 3-dimensionality of it. This was done, again, with Carolyn Larabella, who I mentioned earlier, and Mark Legros. And this is this water window just below the absorption edge, a little bit longer wavelength, a little lower photon energy than the absorption edge of oxygen. So the water is not absorbing, and this has been cryofixation, okay, has been, okay, and this was a yeast cell. And here is one of those yeast cells. Maybe we have it here. No, the next, well, here's some of the details. Okay, again, no use having a 10 nanometer zone plate because of the depth of field limitation. Here we go. So this is a yeast cell, and this big thing is a vacuole. The purple thing is the nucleus, and these little vesicles, X-ray absorbing vesicles that contain protein. This gigantic vacuole is eating up the inside of this yeast cell, and so this yeast cell was about to die. It was about to burst because all the contents was being eaten by the vacuole. But if I go, it won't let me go backwards. Yeah, this is showing the problem here of the lateral resolution and the depth of field. Okay, and the depth of field was only a few microns, but the object is about 10 microns, so that limited the resolution. It says 60 nanometers here, but it wasn't that bad actually. But it's like 30, 40 nanometers. So here it is again, rotating. So there's a three-dimensional reconstruction. So what happened was that this thing was rotated. As I believe with this particular image, they took an, with this particular cell, I think they took an image every 2 degrees or something like that. Okay, and they got this, and then from all of those angular views, not a complete 180 degrees, but close, they were able to do the reconstruction. So so Carolyn Larabelle now has her, she's a professor at UC San Francisco, and she's also at LBL, and she and Mark Legros built this microscope of their own, just for tomography, for biological imaging. These are their support, NIH, DOE, and the Gordon and Betty Moore Foundation. So they're quite successful. If anyone wanted to look them up, they're out there. And Carolyn Larabelle is just wonderful to talk to. And Mark is very, very helpful in explaining things and building things. This is an image that, this is from tomography. It was done by Gerd Schneider at Bessie and Jim McNally at the National Institutes of Health when Jim was at the National Institutes of Health. This is one slice of a reconstruction. So this was done, again, just in the water window, 25 nanometer outer zone width, 1 degree intervals covering a total of 120 degrees. The total exposure for all images, 5 minutes, okay? And you can see some of these things. This is the nuclear membrane, which you can see is a double membrane. And you can see mitochondria, endoplasmic reticulum, some of these vesicles I was telling you about that are used to carry proteins around and through the wall, etc. And here you're seeing slices. After the reconstruction is done, you can go through the object. Okay, so I think we'll hear more about that from Hannah. So here now is, here's now shadow casting and tomography. This again is tomography rotating the sample. It says 1,500 projections, so there's quite a few projections at 20 kilovolt X-rays from a bending magnet, actually a strong super bending magnet, so they get hard X-rays. And you know, the critical energy is 11 kilovolt, but remember, you can go out to several E-crit, 4 E-crit, and you still have decent flux. So they've gone beyond the critical to 20 kilovolts, and this is at the Swiss light source, okay? And so they use bending magnet radiation, double crystal monochromator, and then those X-rays with only a small angular spread coming off the crystals, because that's all, they will only reflect high reflection at a very small angle. And then here's some sample that they're going to scan, and there's no optics here. So they're not going to get down to nanometers because there's no optics to do that. They're just shadow casting with a little bit of a blur, but they can get a micron or approximately a micron in resolution, where now these the shadow of this object, the X-ray shadow, hits a scintillator. It's a material that you illuminate with X-rays on one side, and out comes visible light on the other side, just like a shadow of it. And then you capture with a microscope objective, which could provide you with a resolution of a micron or a half a micron, something like that. And it images that shadow cast back onto a visible light CCD. And they have done tons of information. These people are also working to do clinical work where they replace the Swiss light source with an X-ray tube, commercial X-ray tube, and maybe use one of these grading-based interferometers or grading-based imaging to do this. But in many cases, they simply do shadow casting. And this is an example of work which they did imaging fossilized embryos, which are obtained from the desert in China, where they're finding all kinds of interesting material from this period of around 500 million years ago, which was a period where new life forms were appearing rapidly. And these things are a few millimeters. You can see the scale is 100 microns. And these are fossilized embryos. And so people were interested in saying, what did early, was this a dinosaur or what? I don't know. But what does the developing embryo look like on the inside? And the technique, this is the technique that works. If you try to slice them, they just crumbled. So, okay, they did this. It says 17.5 keV, not quite 20 keV. And this was published, it was a cover of Nature. Donahue was the I don't know if you call him the anthropologist, but okay, and Marco Stampanoni, this is his development. And it's Marco who continues to lead that effort. He's now a professor at Zurich, but he also leads many efforts to develop clinical applications where they replace the synchrotron, but they use some of the other techniques. Oh yeah, so here's the same thing. Okay, oh, and it says the resolution is about a micron and a half. Okay, so there's a development where you don't need X-rays, you don't need optics, X-ray optics, but you can get really useful information. So not everything in the world is nanometer scale. There's lots of micron scale and even tens of microns and hundreds of microns, different communities. But here is an example at the micron scale. Oh, and the same group has looked at all the blood vessels, the architecture of blood vessels and veins and all of these other things for a mouse, okay? A mouse brain, okay? So again, they've rotated this thing and then done a CT reconstruction. This is something completely different. So this is part of the studies at ESRF led by Marina Cote, who's down here. So she's at ESRF, and she started this field, really, of looking at art, not only paintings, but vases and all kinds of structures from thousands of years ago or hundreds of years ago, and analyzing them. What kind of paints did they use? Where did they get these paints from? If they were discolored, how to bring back the color to them. And so she started this herself, I guess 10 years ago, and now she has done so many interesting things. She's in the news a lot, and she's certainly the leader of a new field, and she works with tons of people at museums of different museums in France and Spain and other places. And this is the scheme that she had, which she developed the expertise for the physical parts of it. The microscopy would have come from Jean Sussini, who was her boss at the time, and he's now deputy director, I think, of ESRF. And this is an example. So this is their scheme: undulator, double crystal monochromator, maybe an optic, maybe just shadow casting. If there's a zone plate optic, they need an autosorting aperture. Okay, maybe maybe then look at diffraction out of the back to see diffraction. So they slip in a diffraction camera or something else. And maybe they look at fluorescent emissions. They have all kinds of capabilities. This was one of her early successes, this 18th century, I'm sorry, 18th dynasty, not century, Egyptian glass vase, which is from 1500 BC, and it maintains these beautiful dark and light blues. And how is that possible? And so Marina did fluorescence studies. She managed to get the museum to give her a little flick from the vase and then illuminated it and looked at the fluorescence. And from that, they were able to figure out what was the chemical content that was giving the color. And it turns out to be an early study of nanostructures. So certainly the Egyptians in 1500 BC were not aware of nanostructures, but they were making some nice mixtures, which they saw held the color very well. So, and that's a story that went on around the world. In Turkey, if you go to the Hagia Sophia, you'll see also incredible blue, also maintained there. It doesn't fade with thousands of years because it's atoms and molecules, and it's not some paint. So yeah, so this is Marina would do, for instance, sulfur, and here chlorine, looking at different absorption structures. You see here in KeV, and learning to identify, making a library of absorption features, spectral X-ray absorption spectroscopy, XAS. Okay, and looking at that and looking at all this structure and then using that to identify unknown objects, unknown paints or other things. Okay, this is one of her famous things, this, what does it, where there's this, I can't see the title, it's blocked on mine, but there's this famous painting of this grass, and what she found was that behind it, the artist had done this painting of a woman. Okay, and it turns out he did many of these paintings of women, and some of them became famous and others got painted over, and they found all of these things. Okay, and so here it says they're using X-ray absorption near edge structure. Okay, oh yeah, so this is a Van Gogh. A Van Gogh's painting. I guess the title is up there, but I don't see it. Okay, and this is showing his painting versus, because they're wondering, well, what paints could they use in those days? Okay, and comparing it to available things. And this is just showing, identifying the materials they used, the paints, what elements were used in the paints. Okay, and where do they come from in the periodic chart, and what kind of objects have they looked at in her lab over all these years. Okay, so we still have some time. So this is, lens is not necessarily needed, not only to do shadow casting, which would give us resolutions in the micron range, but here's a case where they're trying to get nanometer resolution, even get beyond 10 nanometers, get to 1 or a few nanometers. So the technique here is called CDI, coherent diffraction or diffractive imaging, where coherent light, undulators with a pinhole, or perhaps a free electron laser, illuminates some object, and there's a diffraction pattern produced by this object and all of this messy features that it has inside that the scientists are trying to understand what resolve those features, and they're looking for, as I said, resolutions they want to get down into below a nanometer if they can. So what this technique does is it just records intensity, but what they try to do is do an iteration here where they take this intensity and back propagate it and see what they get in the plane back propagate the recorded intensity pattern to this plane. And when they do that, they'll see some structure around the object, but they'll also see some structure, reconstructed structure that's out here, because they didn't have the phase, they didn't have the electric fields here. They only had the intensity, so amplitude but no phase. So in the reconstruction, it's not accurate. And they wind up seeing some features where the object should be, but see a lot of features outside. And so they say mathematically, well, we're not supposed to have anything outside the object. In fact, they'll have an absorption defining aperture around it, and they'll say, okay, that radiation that's reconstructed there shouldn't have been there. Let's block it, and then after we block it, let's numerically reconstruct it again and see how the pattern here changes. And when they see the changes here, then they say, okay, let's back propagate that again this way. And again, they still see some structure outside, so they block it again. And they do this iteration over and over again, and maybe thousands of times, okay? And one of their goals is eliminate the zone plate, not only because of resolution, but also inefficiencies, but it's the resolution. So this is the technique: illumination, some aperture not shown, recording of the diffraction pattern, and then numerically go back and forth from this plane to this plane, back and forth. And so that's called coherent diffractive imaging, CDI. And here's some early results. These are 50 nanometer gold balls, okay? They were mounted on a pyramid structure, okay? This is the photon energy. This was done at the ALS, but this became a big thing, say a decade ago. It was spread like wildfire around the world, because all of a sudden, you didn't need to be able to make a high resolution zone plate lens. All you needed was a good computer with lots of recording space. So anyway, here is a reconstruction, and when you look at this closely, you can see these 50 nanometer gold balls. In fact, here, you can see a whole bunch of them here, okay? This is a 500 nanometer scale. So this was work by Henry Chapman and Stefano Marcassini and others, and they're still at it. They do this a lot, but they've evolved. They still do CDI, but they've evolved into what I call tychography, what I mentioned about tychography, so we'll have some references on that. Anyway, so this was an early demonstration that it works. This is work that was done at LCLS. So this is again CDI of single molecules, okay? And at LCLS, and here's the nominal scale, okay, the photon energy, etc. And this is led by János Hajdú at that time. János Hajdú and various postdocs who went on to great positions after this. And they're mostly from Uppsala at the time, but this again spread like wildfire. But this was a state of the art 5 years ago, 2015. And so for instance, this is the recorded diffraction pattern. This is the reconstruction of that diffraction pattern after many of these iterations. And here is numerically what a Numarsky microscope image would look like of that object. And the reconstructions here, you see that, for instance, this is a micron, 1,000 nanometers, and the little white circle here is what they think was their spatial resolution here. So these are all sort of like, you see, 80 nanometers to 100 nanometers resolution. So not where they want to be, but these are brave pioneers who are trying to develop the technique to get to molecular resolution imaging, so that they can look at, as it says, isolated proteins, viruses, etc.
Professor?
Yeah.
Since the wavelength obviously affects the resolution,
For sure.
I may have said this earlier, but how does this wavelength compare to like the initial shadow casting experiments?
So in the ones I showed you, mean like the rotating sample at Swiss Light Source, they were getting a resolution of a micron, and they were using 20 kilovolt it said, remember, just under 20, 17.5, it said in the slide. So their wavelength was much shorter. These people would now be working not in the soft X-ray region, but they would be doing the same work. If I had a more modern resolution, they would probably be working at, I guess, around maybe 10 keV or something like that, where the wavelength would be shorter. So here the wavelength is a few nanometers, 2 nanometers, 2.5 nanometers actually. And so, but the resolution, that's not what's limiting the resolution here. Okay. So this is a continuing area that's done at all the big X-ray FELs. I don't think they would be doing it anymore in the soft X-ray region. Thanks. So this is tychography. So CDI, basically people were finding that with CDI, going around these iterations from plane to plane and back propagating, there would be closure wouldn't always happen around some believable structure. Many times it would, but sometimes it wouldn't, and you really didn't have a good handle on it. So people were looking for a better way, something better than CDI, and what the community came up with, some people in the community, and in particular at the Swiss Light Source, was what's called tychography, where they use a Stixum microscope. Let's say it's shown here as a hard X-ray, but it might just as well be soft X-ray, where there's an undulator, a pinhole spatial filter for coherence, a double crystal monochromator or a grading monochromator for soft X-rays, a zone plate, the order sorting aperture, and then put that on the target. So in Stixum, you'd want all of this to produce the smallest possible spot size, and then you'd do your raster scanning. But in tychography, you don't want to get to the smallest possible spot size. You want it to be a little bit big, and then when you raster scan, your images will have a certain overlap, and that overlap will provide a redundancy in the reconstructions, and the reconstructions will be more accurate. So the big thing about tychography is it introduces a redundancy by having scanning for small pixels, but with a little overlap. So in each reconstruction of each little pixel area has overlapping information with the next one. And you use that, or they use that, to get higher resolution and more accurate reconstructions, okay? A big problem with this is the data collection time is very long. Yeah, I don't remember if it's an hour, but I think it is actually something like an hour. So for the new diffraction limited storage rings, for instance, at the ALS, they're going to cut that down by a factor of 100. At the hard X-ray facilities like APS, they won't get a factor of 100, but I think they get a factor of 30. So it's going to be an enormous improvement. And yeah, and here are some images. These images were this microscope at the ALS, the tychography at the ALS. The instrument development was by, for those of you who know him, Khazra Nawrouzi, who was an this was his PhD thesis for ASNT, and he was part of the ALS team. And later his boss, Shapiro, who was the leading behind the whole team, continued this effort with others, and they basically got this down to a resolution. I think when Khazra was there, the resolution was a world record at about 7 nanometers, and I think now it's more like 5 nanometers. And so around the world, there's only a few that are really doing high resolution tychography, Swiss Light Source being perhaps the leader. Okay, at any rate, so that's a second kind of basically what they would call lensless imaging, even although you need a lens to do the illumination, but there's no lens in it. So just some comments then on diffraction limited storage rings, which we've talked about before. We don't need to read this, I think, but basically the idea is that you would take an elliptical electron beam cross-section and reduce it to, right now, if we have these elliptical high eccentricity ellipses, and we throw most of it away. We only use the little part in the middle that goes through our pinhole and sets the spatial coherence in both directions. So in the vertical direction, we're kind of close, but in the horizontal direction, we're typically off by 30. So with the new ellipse at the ALS, this 30-to-1 ellipse is going to be brought down to a circle. So there's a factor of 30 improvement. All the photons that used to be out here are now going to come through the middle. It's not like they're being thrown away. It's just that the electron beam coming through will have a circular cross-section, and all of that radiation can be focused through the aperture, through the pinhole aperture. So there's a factor of 30 there, and there's another factor of 3 that comes from slightly increased current, a slightly different undulator that has certain performance advantages. You know, the coherent part of undulator radiation goes as n squared. So if you had 89 periods in one of the common ALS undulators and you went to 270, it goes as n squared, so you get an extra factor there. So that extra factor of, let's say, 270 over 89, so that's like, maybe that sounds like too much, but anyway, there's some factor by having longer undulators and getting an n squared effect, slightly increased current, and a factor of 30 in reduction of the ellipticity. So for the ALS, it's going to be a factor of about 100. And that factor, by the way, was in homework problems I didn't give you in the synchrotron chapter. So what are what's going to gain by this? Well, basically, certainly all the ones that use coherent radiation. So the STIXM, the scanning transmission X-ray microscope, the CDI, the tachography, certainly they're all going to gain. They need coherent radiation. But in fact, all of the imaging, even nanoprobes that get micron resolution and the full field microscope, which doesn't require coherence over the whole thing, what's going to happen is that when the ellipse closes, a lot of radiation, which we now throw away, is going to come through. So all of the imaging systems are going to gain. And this is just some list of a couple of them, but it's just what we said in the slide. And so for instance, an ALS, this is an ALS beamline, would gain between 50 and 100 depending on which undulator. So the optimum undulator would gain about 100 times reduction in exposure times, okay? Maybe 50 times in some different undulators. And the same thing for argon. Their total reduction is going to be a factor of about 30, but in some of their experiments, it's absolutely crucial that they get that factor of 30. Okay, and this is spring 8. This is the hardest X-ray undulators in the world. These are the old curves, but these curves are also going to all show increases in coherent power of order 30. Okay, and this is the end of the lecture. So how are we doing on time? So this is good. We have 5 minutes before the kids from HKN are going to come and see if they can coerce you into making out these reviews. But anyway, so what about comments from you now on anything today or anything about the projects? Or I guess I can shut this down. Hey, there we go.
We're still doing 3 days of project presentations, correct?
You're correct. In fact, I have it here in front of me. Let me just see if I can get rid of this. Why can't I get rid of this?